This video is about leukocyte activation and migration. So, this particular study note is developed by APJ Patcharaki, Government of India, MHRD. So, I would like to express my sincere gratitude to the development team for developing the study note. Leukocyte activation and migration. By the end of this chapter, students will be able to appreciate that migration of leukocytes is a critical component of both innate and adaptive immunity, as the immune responses primarily depend on the capability of leukocytes to traffic from the blood circulation into the tissues. The foremost objective is to discuss the mechanisms, profile, and consequences of neutrophil recruitment, lymphocyte re-recruitment, monocyte trafficking, all of which require adhesion and transmigration through blood vessel wall that has been stimulated. The pathway that helps facilitate the movement through the early phase of leukocyte or epithelial cell when it comes in contact with the lumen of vessels will be discussed, especially about migration across the perivascular basement membrane, a hurdle that is rarely explored in the millennia of leukocyte migration in vivo. Introduction. The phagocytic activity of leukocytes, together with its ability to differentiate into antigen-presenting cells and monocytes, contribute in innate and adaptive immune response. Leukocytes are derived from the progenitor cells in the bone marrow and are in blood circulation as monocytes, but segregate as dendritic cells of the myeloid origin in the periphery and in the tissues as macrophages. Therefore, a critical component of both innate and adaptive immunity is the migration of leukocytes to inflamed injured sites, because the immune response depends primarily on the ability of the leukocytes to pass from the blood circulation to the tissue. The threat signals triggered by inflammation injury stimulate the resident cells of the innate immune system, the mast cells, macrophages, and dendritic cells that cause secretion of cytokines and release of other pro-inflammatory mediators to stimulate neighboring endothelial cells in the blood vessels. Henceforth, a cascade of events is elicited that enables the circulating leukocytes to distinguish the vascular endothelium in the inflamed tissue and to interact with the blood vessel wall. To achieve this, precisely coordinated pathways are already present that recruit intravascular leukocytes and assist them to infiltrate the vascular wall, allowing them to migrate to the injury sites or to infection site without causing any otherwise damage to the vessel while emigrating. The foremost aim is to discuss the profile, mechanism, and consequence of recruitment of neutrophil, lymphocyte recirculation, monocyte trafficking, all of which necessitate adhesion and transmigration through stimulated blood vessel wall. The mechanism that facilitates the initial stage of leukocyte endothelial cell interaction within the blood vessel lumen will be discussed, in particular migration through the perivascular basement membrane, a barrier that is rarely investigated in the context of leukocyte emigration in vivo. First, why do leukocytes migrate to the tissue? Leukocytes originate from the bone marrow progenitor cells found in circulation in the blood as monocytes and differentiate as myeloid dendritic cells in the periphery and into the tissues as macrophages. Cells of the immune system constantly traffic throughout the body to justify their function of immune scrutiny, leading to pathogen elimination, like blood leukocytes, which comprise lymphocytes, monocytes, dendritic cells, and neutrophils, cross the blood barrier and travel to the infection site. The phagocytic activity of leukocytes, coupled with their ability to differentiate into APCs, contribute to the innate and adaptive immune system. The third signal caused by injury inflammation trigger the innate arm of immune system, like macrophages, mast cells, and dendritic cells secrete pro-inflammatory cytokines to stimulate the endothelial cells. An important component of both innate and adaptive immune system is the migration of leukocytes from circulation to the tissues. Importance of cell adhesion molecules. SCAMs are required to ensure that circulating leukocytes migrate to the inflamed tissue or to the peripheral lymphoid organ, and it is important that the cells adhere to and cross over the endothelial cells lining of the blood vessel. Extravasation. Endothelial cells express leukocyte specific cell adhesion molecule. CAM present on the leukocytes help them to attach to the endothelial cells of the blood vessels to enhance the strength of the interactions among the cells of the immune system, THAPC, BHB, CATL target cells. Naturally, CAM can be classified into 4 families. Mucin-like CAM, which express sialylated aldehyde side chains, in an integrin family, which is the heterodimer consisting of alpha and beta polypeptide, then selectins, wherein L-selectin is expressed on the leukocyte, and P and E-selectin are expressed on the inflamed endothelium, which expresses CD62 that ends in a lectin domain. Chemokines facilitate both activation and migration. Are small polypeptides mostly made up of 90 to 130 amino acid residues modulate the chemotaxis, adhesion and activation of leukocytes, regulators of leukocyte trafficking. Few are principally involved in inflammatory response. Some are constitutively expressed and have an important homeostatic or developmental role. Chemokine-mediated effects may not be restricted to the immune system. The pro-inflammatory cytokines are induced in response to infection and are capable of recruiting phagocytes and lymphocytes to the inflammatory site. Multi-step model of leukocyte and neutrophil recruitment. This multi-step model of leukocyte transmigration includes tethering to slow rolling and regulated rolling of leukocytes across the vascular endothelium. Graphic representation of the sequential steps involved in leukocyte migration through the stimulated vessel walls indicating the capture for transitory adhesion, rolling, slow rolling, firm adhesion, diapedesis or transmigration across the endothelium. Multi-step model. The adhesion cascade includes capture, slow rolling, arrest, adhesion strengthening, intraluminal crowding that involves spreading, paracellular and transcellular diaperesis, and migration through the basement membrane. Under normal physiological conditions, the lining of the endothelial cells of the blood vessels exhibit an anti-adhesive and anti-thrombogenic surface that ensures blood flow along with the blood components to maintain homeostasis. So, under normal physiological conditions, the lining of the endothelial cells of the blood vessels exhibits an anti-adhesive and anti-thrombogenic surface that ensures blood flow along with the blood components to maintain homeostasis. The neutrophils move slowly, rather rolls over the endothelium, unlike the other blood components that move along blood flow with a high velocity. But at the injury site or the place of infection or inflammation, presence of endotoxins, expression of certain cytokines and other secreted molecules renders adhesive properties to the circulating leukocytes, thereby initiating cascade of leukocyte emigration. The neutrophils move slowly. But the injury site or the place of infection or inflammation, presence of endotoxins, expression of certain cytokines and other secreted molecules renders adhesive properties of the circulating leukocytes, thereby initiating a cascade of leukocyte emigration. Then area of inflammation. Here we could see slow movement of neutrophils in the blood vessel. Then rolling. The responses are generally mediated by weak and revocable molecular interactions between the selectins, P-selectin, E-selectin and L-selectin present on the endothelium and their counter ligands, PSGL1 present on the leukocyte. The interaction between selectins and PSGL1 facilitates rolling. PSGL1, P-selectin, ICAM, IL-8. Integrin activation. The active upregulation of P-selection in inflamed endothelial cells reduce the velocity of rolling leukocytes. In addition, certain integrins such as VLA4 can also mediate leukocyte rolling under conditions of flow. The slowing down of leukocytes during rolling facilitates the ligation of specific and high affinity G protein coupled receptors by endothelial cells of lumen associated activating factors such as chemokines. This interaction augments the ligand binding profile of integrins that is achieved by increased affinity and avidity of the molecule. Integrin activation plays a critical role in facilitating the adhesion of leukocytes firmly to the endothelial cells, leading to flattering of the leukocytes over the endothelium, thereby ensuring the two crucial steps in leukocyte emigration. Beta 2 and beta 1 family of integrins are the major integrin involved in this step of leukocyte or endothelial cell interaction. Firm adhesion. At the sites of inflammation, interleukin 8, IL-8 molecules are secreted by the endothelial cells and remain in the periphery of these cells bound to heparin sulfate proteoglycan. This leads to conformational changes that result in the formation of high affinity interactions between the ICAM molecules on the inflamed endothelial cells of the vessels. These strong interactions assist the leukocytes to firmly adhere to the surface of the endothelial cells. Interaction of leukocyte with the vascular endothelium is a highly coordinated process that includes the arrest of free-flowing leukocytes in the blood with subsequent leukocyte rolling arrest, firm adhesion and ensuring diapedesis. These interactions happen only under high shear stresses within the venules and depend on multiple families of adhesion molecules. As a response to infection mediators, leukocyte gathering is generated to be crucial for an adequate defense of the host organism to any kind of injury or infection. Activation of endothelium contributes considerably to systemic inflammatory response to septicemia and enhanced expression. Within the setup, leukocyte transmigration through vessel walls of postcapillary venules is the concluding stage of a cascade of response of leukocyte mediated by a sequence of molecular interactions that primarily mediate the slowing down of velocity leukocyte rolling, followed by firm adhesion of leukocytes to the endothelium and finally transmigration to the vessel wall. Leukocyte transmigration acts as a means of guiding the emigration of leukocytes from the lumen of vessels to the extracellular vascular tissue, thereby playing a crucial role in monitoring the phenotype of the emigrated cells in such a way that responsiveness of behavior of leukocyte to chemoattractants directs the migration and interaction with the molecules of the extracellular matrix or extravascular tissue may be further regulated. Therefore, such a mechanism clearly contributes to the capability of immune system to elicit an appropriate, optimal and localized tissue response to a variety of extravascular stimuli without triggering any non-specific damage to the vasculature. Leukocyte migration requires polarization. Leukocyte migration requires polarization. Leukocytes that adhere tightly to the vascular endothelium need to breach this vascular barrier to reach the underlying inflamed tissue. This can be achieved by active integrin-mediated cell migration. Migration requires polarization of the cell body and the formation of a lamellipodium. It also requires the formation of uropod at the tail of the leukocyte. The formation of this structure is driven by integrins and the cytoskeleton. Nonetheless, in situations of uncontrolled infiltration of leukocyte that may exceed either in duration and or magnitude or incogress in its location, transmigration induced alteration in the phenotype of leukocytes may lead to stimulation or exacerbation or deleterious inflammatory response in the host. Then, transendothelial migration of leukocytes. Finally, the basic steps for intravasation should include interstitial migration towards the vessel, migration across the basement membrane and endothelial barriers, diapedesis, and ultimately release of luminal leukocytes into the circulation. It is now clear appreciated that diapedesis, if it occurs during intravasation or extravasation, can ensure two distinct routes of pathways, either via disassembly of the intracellular junction to form a paracellular gap, paracellular diapedesis, or via the formation of a transcellular pore directly through the individual endothelial cells, transcellular diapedesis. The concept of one cell passing through or in effect entering another cell, or as occurs in intracellular diapedesis, might seem a bizarre and unlikely one. The complete migration of the leukocytes through the vessel wall into the surrounding connective tissue is called the transendothelial migration. Transendothelial migration through endothelial barrier, diapedesis. When integrins are activated and the cell migration machinery is functionally polarized, the leukocyte transfers along the luminal side of the vascular endothelial cells and then transmigrates primarily by transiting between neighboring endothelial cells. PFCAMs. Activation of PFCAM CD31 facilitates the movement through the endothelial junctions that are subject to constant remodeling to enable leukocyte trafficking. Nevertheless, leukocytes have the ability to pass through the body of the endothelial cells too. Although there is a prevailing transcytotic pathway for extravasation, it may not be the crucial route often taken by leukocytes. A leukocyte taking the conventional paracellular route faces the vascular cells connected by a large array of endothelial junctions which need to be penetrated. These junctions preserve the integrity of the endothelium and monitor vascular permeability. Three types of endothelial junction have been described: tight junction, adherence junctions, and gap junctions. The endothelial junctions resemble those found in epithelium, but their three-dimensional organization is often disordered, indicating the perpetual remodeling that commences from leukocyte trafficking. Undeniably, transmission is a vibrant process in which the tight junction, gap junction, and adherence protein facilitate the final step of leukocyte extravasation, functioning as liquid engulfing transmigrating cells rather than as a gate. When there is a lack of inflammatory trigger, the adhesion molecule along with chemokines that are involved in collection of macrophages and DCs in peripheral tissue should be constitutively present. The DCs and macrophages originate from the bone marrow and hence are required to be recruited from the circulating precursors in the blood. Among us, the constitutively expressed chemotactic molecules that act on circulating monocytes as CXC chemokine, commonly identified as breast and kidney expressed chemokine BRAK, has been projected to the involvement in the constitutive tracking of monocytes. However, the role of CXCL14 or other chemoattractants such as deficins have not been explored with regard to the recruitment of monocytes to non-inflammatory tissue. T lymphocyte recirculation occurs via multi-step pathway. The maximum exploratory behavior among cells is displayed by T lymphocytes. They are capable of migrating throughout the body and are able to explore most of the tissues. Beside large-scale displacements, T lymphocytes continuously probe their microenvironment in a mode preferring recognition of specific antigen at the surface of antigen-presenting cells. Mobility is a characteristic property of T lymphocytes that promise timely and optimal recognition of extragenous antigen at the site of their antigenic challenge. After stimulation by chemokines, adhesion to the specialized endothelium will occur only when upregulated integrins interact with ICAM molecules expressed on the endothelial cell. In the lymphoid tissue, the glycoprotein CD34 displayed transiently at the surface of endothelial cell forms a momentary interaction with L-selectins. Transmigration. When T lymphocytes travel across venular blood vessel walls to enter various tissues and organs, it is called transmigration or diapedesis. The ability to migrate is crucial for the T lymphocytes at all the life stages, from entry of T cell precursor into the thymus for T cell expansion to entry of naive T cells into lymph nodes for activation, migration of effector T cells into tissues to combat infections. Similarly, transmigration remains a multi-step cascade that comprises lymphocyte rolling, signaling firm adhesion, and transmigration. Each step is constantly regulated by interaction among adhesion molecules and chemokine receptors that are expressed on the surface of lymphocytes and their ligands that are correspondingly expressed on the endothelial cell lining the vascular wall. Lymphocyte circulation is quite similar to the leukocyte extravasation, but homing receptors direct the migration of lymphocyte subsets to particular lymphoid tissues or inflamed endothelium. Lymphocyte rolling. The contact between lymphocyte and endothelium is initiated by lymphocyte rolling that is mediated by repeated binding release events between selectins and their ligands. During the process of rolling, chemokines are displayed on the surface of the endothelial cell that can interact with chemokine receptor on the lymphocyte surface. Then signaling. Simultaneously, interface between a chemokine and its receptor results in a cascade of intracellular signaling that may further activate the adhesion molecules of the integrin family to translate the signals. Then firm adhesion. When the integrins are activated, the lymphocyte will display high affinity binding sites that interact with the cell adhesion molecules on the vascular endothelial cell, resulting in slowing of lymphocyte, its arrest and strong adhesion. Transmigration. The method of transmigration is not very well characterized as compared to other steps, especially in terms of T lymphocytes. Tissue specificity of T lymphocyte transmigration. Transmigration does not occur indiscriminately but is highly regulated to enable the T lymphocyte to precisely enter the specific tissue. Since the blood venules present in different tissues exhibit a discrete arrangement of adhesion molecules and their corresponding chemokine ligands, only T cells that express an accurate combination of chemokine receptors and adhesion molecules will be allowed to enter into the specific tissue. For example, the chemokine CCL25 and CAM-MADCAM1 are involved in recruitment of T cells that express the chemokine receptor CCR9 and integrin A4B7 to the small intestinal mucosa. T cell migration is generally mediated by cytokine integrin activation, which directs the cytokine to provide critical guiding signals to direct T cell migration to specific tissues within the body. Evolving substitution supports substantiation supports that activated T cells migrate to certain vascularized tissues without chemokine receptors or with the inhibition of chemokine receptors. The chemokine independent mechanism generally involves LFA1, through which T cells further activate integrins, induce adhesion and subsequent cell migration. The control of T lymphocyte motility. The control of T lymphocyte motility is fundamental to the multi-step method of extravasation from the bloodstream into tissues. The mechanism of tethering, rolling, firm adhesion, trans-endothelial migration has been discussed remarkably on interaction with endothelial cells. T lymphocytes crowd with an antibody motility. Amoeboid motility. The shear stress exerted by blood flow contributes to the creation of LFA1 dependent adhesions. The migrations of T lymphocytes is oriented towards blood flow as they interact with the innermost surface of blood vessels. The mechanotaxis of upstream flow relies on passive self-steering mechanism. Lymphocytes exhibit dynamic protrusions during transendothelial migration. The exploratory structure rich in actin and are viewed as podosomes or philopodia depending on the state of activation of lymphocytes. Apparently, they allow the lymphocytes to scan the endothelial cells and to ascertain areas that may be favorable for transcellular lipidasis. The microenvironment of the endothelium also determines the various requirements of the T lymphocytes. This is clearly shown by the role of uropoid contractibility in transendothelial migration to access the lymph nodes but does not reach the bone marrow. Monocyte trafficking. Monocytes play a fundamental role in tissue homeostasis, defensive immunity, and both enhancing and reducing inflammation. Several of the functions of monocytes are carried outside the vascular compartment, hence migration and trafficking are necessary. Monocyte recruitment. The recruitment of blood monocytes to the site of injury or infection and their diapedesis through the endothelium, also called extravasation, are critical events at the onset of inflammation, followed by differentiation and successive downstream cascade of the inflammatory response. A tightly regulated multi-step process consisting of a series of interaction between endothelial cells and cells of the immune system precedes the actual transmigration step. For monocytes, this progression is basically believed to track the cascade paradigm initially described for neutrophils. Transient activation of the endothelium is initiated by inflammatory chemokines such as tumor necrosis factor or IL-1B, which originate from the tissue macrophages. After chemokine stimulation, the adhesion of monocytes to the endothelium occurs only when activated integrins interact with ICAMs. They elicit a quick expression of adhesion molecules such as E and P-selectin, intracellular adhesion molecule ICAM, and vascular cell adhesion molecule 1 VCAM, and the presentation of bound chemokines on the luminal surface of the endothelium. The selectins interact with the O-glycosylated carbohydrate ligands that are displayed on P-selectin glycoprotein ligand 1 that are constitutively expressed on all monocytes. This interface allows the monocyte to roll slowly on the endothelium and balance the high shear stress applied by the flow of blood in the vessels. PSGL1 expression is significantly higher in inflammatory monocytes than by resident petrolling monocytes that facilitates their adhesion to atherosclerotic lesion. So LY6 monocytes extravasate into blood in a CCR2 CCL2 dependent manner. This monocyte emigrate into blood via CX3CR1 CXT-CL1 signaling. LY6-CHI in blood can perform different functions. They can phagocytose microbial pathogens and migrate to inflamed tissue sites. Atherosclerosis is categorized by the upregulation of endothelial adhesion molecules like P and E selection and VCAM1 in lesion-prone areas. Deficiency in P and E selection as well as ICAM considerably reduces the size of atherosclerotic lesions in ApoE2 mice individually. In addition, in conditions of inflammatory response, monocyte rolling largely depends on monocyte expressing very late antigen 4 and CD44. The endothelial adhesion molecule VCAM1 binds to VLA4 and facilitates slow rolling on cytokine activated endothelium, thereby ensuring the switch between rolling and firm arrest. The firm adhesion of monocyte to the endothelium is synthesized by CC and CSC chemokines, CCL2 and IL8. Monocytes may also use VLA4 for firm adhesion. Monocyte arrest is followed by a directional chemotactic and mechanotactic step in which the monocyte spreads, popularizes, and consequently locomote alongside the epithelium to find desired sites for extravasation. This intraluminal crawling depends on the leukocyte integrins, lymphocyte function associated antigen 1 and macrophage 1 antigen, as well as the endothelial ligands ICAM1 and ICAM2. By blocking these adhesion molecules, the crawling and the subsequent transmission were restricted. This step involved probing the apical surface of the endothelium with the monocyte membrane protrusions called lamellipodia. It is important to understand that this crawling is markedly distinct from patrolling, which is definitely intravascular and does not lead to extravascular or in steady state. Finally, the monocytes have to negotiate with the endothelium to exit the blood vessel and lamina basalis and the embedded pericytes. Transmigration seems to be the central event on the monocyte adhesion cascade, as the only step that is never reversed. After extravasation, the monocytes differentiate into macrophages. Two phases of monocyte transmigration across the vascular endothelium. The thin monolayer of epithelial cells to constitutes the primary physical barrier between blood and tissue. Endothelial cells have no true tight junctions. Adherence junctions between the cells and integrin and cadherin anchors to the basement membrane from a network that tightly regulates vascular homeostasis and restrains leukocyte transendothelial migration. Three types of migration of monocytes have been observed: intravasation, extravasation, paracellular diapedesis, transcellular diapedesis. Monocytes prefer transcellular migration in only 10 to 30% of events. Strong activating stimulus increase the overall occurrence of transendothelial migration in monocytes. Live cell visualization in vitro and live imaging through confocal intraocular microscopy have greatly advanced our understanding in this field. And extravasation, the predominant paracellular route that leads monocytes through the junctions between epithelial cells and requires junctional remodeling. A different route directly through fusing vesicles and endothelial cell cytoplasm constitute an established mode of monocyte transmigration. Then conclusion. Over the past 20 years, it has become increasingly evident that the innate immune system has a central role in polarizing the adaptive immune system towards T helper cell, Th2 cell, or humoral response. APCs determine the specific recruitment and positioning of T and B cells in the body. We are improving our knowledge of the recruitment of monocytes from the blood to inflammatory tissue and capability of monocytes to differentiate into DCs. For example, new molecules such as RAP1 is its ligand K2 and atypical PKC have been added to chemokine-induced signal transduction cascade that leads to integrin activation and leukocyte migration. Another important discovery has been the involvement of protein complexes in the establishment of cell polarity, which is essential for the directed migration of leukocytes. Furthermore, new adhesion molecules such as JAMS and CD99 seem to be involved in the process that occurs at vascular junctions and function as counter receptors in leukocyte adhesion. Recently described monocyte subpopulations also contribute differentially to mount and efficient specific immune response. Does the constructive migration of monocytes contribute to the establishment of peripheral disease? Is a danger signal required for this recruitment? Is the level of integrin activation crucial for the differentiation and migration of monocyte? What are the microenvironmental factors that control the differentiation of monocyte into disease or into macrophages? Answering these questions will help us to understand the plasticity of the innate immune system and its impact on the maintenance of tolerance.